very elated to be here again for the fourth year with more than 4 years of data to share with you and just to give you an idea of how cover crops are interacting with one of the top input costs for farmers, and that's nitrogen, right? And then how it affects the other input cost, the leading input cost, and that's seed going to yield. And it's really important that we look at this holistically, that we look at it from an agronomic perspective, that we look at it from an environmental perspective, and then economically, how are things working with cover crops. So I'm gonna try to talk about some of that, not so much economics in this particular talk. So I'm gonna be looking at nitrogen application timing, okay? How does cover crops interact, and then how does that impact water quality and production? So we're all familiar with this by now. If you haven't, maybe you don't have the internet or you don't have a cell phone or something, because this has been everywhere, okay? The upper Mississippi River Basin, where we grow the most corn and soybeans to feed the nation. And because of that, we have a system that leaks. We have tile drainage, okay? And nutrients and water move down to the Gulf and causes what we call hypoxia. Okay, that's a fancy word for low oxygenated water, and then the aquatic wildlife in the Gulf then dies. So they call it the dead zone also, that's a nickname. So based upon that issue, which is continuing, and it's not just us, okay, based upon that issue, EPA has mandated every state to come up with a nutrient loss reduction strategy, okay? And that strategy has a target for each state to have a 45% reduction in total phosphorus and nitrate nitrogen by 2025, okay? Now, to achieve this, we can't just look at all of the farmers who apply nitrogen in the spring and say, hey, they're gonna get it done. That's not gonna happen. To achieve this, we have to find multiple methods to increase the efficiency of all nitrogen application programs in the state and across this Midwestern region, the Corn Belt. So we have some data from a survey that we did at ISU with Dr. Spalding and I. And before we got there, from 1993 to 98, Dr. Ken Smikolas had performed a survey, and both of these surveys were basically in the same geographic location. And that's the Mackinac River watershed and the sub-watershed of Lake Evergreen and Lake Bloomington, okay? And so here's how the nitrogen management changed over time. So the average of 1993 to 98, and then we have in the orange the average over a 3-year period from 14 to 16. This is corn and soybean, and then this is the same variable that we're looking at when there's a corn and corn system, okay? What you see here on the bottom is the percent farmers who fall apply, percent of farmers in that watershed, those watersheds who spring apply, percent of farmers that make greater than 2 applications of nitrogen throughout the growing season. So what are you seeing? You should be noticing that compared to the average from 1993 to 98, when you come to 2014 to 16, that there's a drastic decrease in the amount of farmers who are executing fall applied nitrogen. Okay, because of that decrease, you get an increase because they have to put the nitrogen on somewhere to get the full, load, the rate of nitrogen to finish off that corn crop. So because of this decrease, you're getting an increase in the nitrogen being applied in the spring. Another neat thing that we saw, trend that we saw, was that there's an increase in the number of farmers when you compare it back to 93 to 2016 that are actually using nitrogen in a systematic way. Whether it's a percent in the fall, a percent pre-plant, a percent at side dress, or a percent back at planting 2 by 2 with starter, right? There are a number of farmers that are starting to think about this and to be more systematic with their nitrogen application. So then I said, well, let's see if my data in these 2 small watersheds fit with the NASS, USDA NASS producer survey for nitrogen management is saying, okay? And so indeed, we did see a correlation. And this is just in 1 year, but it was across way more farmers than I looked at. So I'm representing about 55,000 acres here, and they're representing way more, as you can see. And so you do have a slight decrease in the fall applied nitrogen from 2011 to 2015, slight increase again in the spring applied nitrogen, and then greater than 50% or a split application of less than 50% in the fall, and then that nitrogen then split out with multiple applications in the spring, whether pre-plant, at planting, or post-planting as side dress has also increased. So we did this on a small watershed. They did this across the state, same trends. So now the big question is, how is cover crops going to impact the efficiency of all of these nitrogen management practices? That's what we need to understand. In order for a farmer to make an informed decision on purchasing and adopting the practice of cover crop and all of the infrastructure you need to do it right so that you don't have a horror story, they need to understand how the cover crop and different species of cover crop will interact with these different types of ways that they apply nitrogen. So basically, when you think about my program in the state of Illinois, you're thinking about the ability of the cover crop to conserve nitrogen, okay? And that nitrogen that it's interacting with, the nitrogen that it's conserving, comes in about 3 pools. Number one, nitrogen that comes from organic matter mineralizing, meaning that it's converting from an organic form like a protein to ammonium and then to nitrate. Not the nitrogen that's applied, but what's in your soil, right? So if you in McLean County, you have a molly soil, what you call black dirt, okay, 2.5 to 4.5% organic matter, if you're treating it right, it's gonna release at least 20 pounds or so, 20, 30 pounds of nitrogen just from the organic matter. Cover crops interact with that. Residual nitrogen, this is nitrogen that comes from that's left over from the last cash crop. So let's say you are corn going into soybean, okay? You're gonna have some nitrogen that's in solution that's left over that the corn didn't actually use. Because really when you look at the literature, that corn plant is only using from 40 to upwards to 60% depending on the hybrid of which you apply. So where's the other nitrogen going? When is it coming out? Cover crops interact with that nitrogen. Then there's the portion of nitrogen that could be applied in the fall. In this region, okay, you're talking about 50 or so percent of farmers still use fall applied nitrogen. You go to Wisconsin, you go to northern Indiana, you're talking about swine manure, okay? And all across the Midwest, there's nitrogen in DAP, okay? Because triple superphosphate is just not that common anymore. So cover crops interact with that nitrogen. So it's absorbing, it's taking that nitrogen and it's putting it into its structure below ground and above ground. And then I studied the release. That's a big question. So yes, it's taking up nitrogen, but when is it letting it go? And then at what stage is it letting it go, let's say for corn growth? Do you get 50% back before V6? Or is it all coming back later on in the growing season at tasseling or VT? Who knows? That's what we're studying to try to figure out, what stage and the timing and the quantity of nitrogen being released from the residue as it decomposes. And that depends on the physiology, whether it winter kills or whether it lays dormant like cereal rye and grows again in the spring, right? Whether it's a legume, grass, or cereal. And then most importantly, these two contribute to the CNN ratio. And that's simply said, the ratio of carbon to nitrogen in the tissue. The higher this number, the slower the release. The lower this number, the faster the release. So if you think about a radish versus cereal rye, which one has the highest CNN ratio? All in favor of radish, raise your hand. This is a smart room. This is a smart room. All in favor of cereal rye, raise your hand. Right, cereal rye has the highest CNN ratio. So that means cereal rye is gonna have a slower, more constant release of nitrogen throughout the growing season compared to radish that's gonna give most of it back very early, almost over 60% before planting. Now, that could be good if you're planting corn into it. That could be bad if you have, let's say a late soybean that you're planting late and you get a lot of spring rain. A lot of that nitrogen we've experienced could leach, okay? So you have to plan. You have to plan based on, what cover crop you have and then what cash crop is following. And then we want to look at the uptake into the actual cash crop, corn and soybeans, right? And then how that affects yield. Okay, so we'll talk about some of that. So at the ISU farm, teaching and research farm, and Russ is here, my good friend, and when I first got here, I didn't know anything about farming. I knew a lot about the concepts, and he just put me in a pickup truck and we just went from implement to implement until I figured it out. Then he let me drive a little bit, not as much though. He said I had to hire students that knew how to drive because I couldn't drive. And so they helped me out at first, and then they kind of turned me loose with the equipment, and we played around with this concept, knowing that there's a lot of fall applied nitrogen in this area, and I'm thinking, nobody's doing research on this fall applied nitrogen to try to increase the efficiency, because really, are farmers really gonna just walk away from it if there's no pressure? I doubt it. You saw the data from 1993 to 98 to 2007 to 2016. There's a decrease, but it's not that much. There's still a lot of fall applied nitrogen out there. So what can we do to make this more effective? So we came up with a green application of fall applied nitrogen, and green meaning that you inject your anhydrous into a living stand of cover crops. We found some amazing things. We found, number one, you see how ugly this is? You think that you're damaging this radish stand? The cereal in this radish stand grew right back over the slits. And in the spring, you'll never know where these tracks are unless you have a guidance system, okay? So that's the first thing we learned. Secondly, we learned that in years of above average temperature in the winter, in the fall, in years of above average precipitation while it's warm, your cover crops are going to interact with this nitrogen and keep it closer to where it was injected compared to if the cover crop was not there. Meaning that if I inject this, and you know the winter we had last winter, right? Very warm, humid, wet. A lot of that nitrogen in the control moved closer to the tile drainage. But the nitrogen that was applied green stayed closer to the surface where it was injected at about 6 inches or so. 6 to 8 inches. So that's one thing that we learned, and we learned that over a 4-year period, okay? When it was cold and it froze over like it's supposed to, like it used to, right? You didn't get a difference between the cover crop treatment and the non-cover crop treatment because the microbes were not as active, and that nitrogen stayed in place, and N-Serve did what it was supposed to do. But at a certain temperature threshold, even N-Serve can't outcompete 7 billion microbes per teaspoon. There's just no way, right? And so you did get some breakdown when the temperatures were too high and it was warm. So we went further, and these are the treatments, and this is where it's located right next to I-55. You may recognize that field right by the farm. That we went further and we developed some treatments, and we wanted to look at a soil health test to see if we could, after about 5 to 6 years, see a difference in soil health parameters, both chemical, biological, and physical, but I'm gonna show you chemical and biological today. So just to brief you, the treatments are a zero control, no cover crop, no nitrogen, okay? Russ didn't like that one that much, but we had to do it because I wanted to see what would happen and after years, what's happening to the organic matter, what's happening to the microbial respiration when you compare these treatments, okay, when you add nothing. Then there's a control, no cover crop with nitrogen. Then there's nitrogen with the cereal alone, nitrogen with radish alone, and then the mixture, nitrogen with the mixture of the two alone. And so all of these plots were managed the same. They received about 180 pounds per acre of fall applied nitrogen. This is all of the nitrogen, worst case scenario, all of the nitrogen applied in the fall. Okay, all of the nitrogen applied in the fall. And so, oh man, this is too little, but I'm gonna explain, okay? So, we decided, we got this opportunity to take samples from those treatments, 3 replications. So every number you see on my charts will be at least replicated 3 times, okay? So it's not a one-shot guess. And we sent our soils to Cornell University, okay, there in New York, and they did a comprehensive assessment of soil health. So let me just break down what this means. They look at the three top parameters of soil health, and that's physical, biological, and then chemical. So in the physical, you have stuff like available water holding capacity, important, okay? That should increase as organic matter increase. Aggregate stability, important, especially if you're on a hill slope, because that decreases soil erosion, okay? And then in the biological, they look at organic matter. They look at easily available soil protein index. And that means simply soil proteins, the proteins that are in the tissue that they know is going to mineralize throughout the growing season. It's easily mineralized. It's not going to convert to your organic matter. It's actually going to mineralize and be released as inorganic nitrogen for the crop to use if the timing is such where your cash crop is growing when it releases. Soil respiration, simple. We all are respiring right now. Microbes do the same thing. And the way we measure that is that we put a measure of soil in a jar. We put a chemical that captures CO2. We top it up, so CO2 moves into the headspace, and then that chemical traps it, and we measure the CO2 in the chemical solution. And we can measure, based on the amount of CO2, we estimate how many microbes are in the soil, and we do division and figure out, okay, you're getting this type of respiration from your soil, right, based on the CO2 that's released. And we did that across treatment. So you can see if one treatment has more respiration, it's an indicator of a bigger microbial pool, okay? Then there's active carbon. This is carbon that's going to turn into dissolved reactive carbon, but it's not yet that, but it's just like the protein. It's easily mineralized, okay? Then the chemical is soil pH, extractable phosphorus, extractable potassium. That's what you normally do with your soil test. And then your minor elements. And we're going to focus on a couple of those. So you see these numbers. They're ratings. They're ratings, okay? So if you see red, that means your rating is below 20 and your soil is not healthy. Not healthy at all, okay? So it's a constraint, actually, to production. From 20 to 40, you'll see orange, okay? That means that you have a low level of functionality of soil health. From 40 to 60, you'll see yellow, okay? That means that you are suboptimal. If you see light green, okay, that means that you are from 60 to 80 and you're excellent. And if you see dark green and you're above 80, that means you're near optimum. You're kind of maxing out on your soil health capabilities. Then they give you an overall score. And they compare your soil to soil samples that they have received from your region, or soils that are similar, okay? So think about the treatments, think about the things that we're measuring, and we're gonna look at what's the impact over 5 years. How many think that the radish alone is gonna have the greatest impact of soil health? Maybe the cereal rye, or maybe the mixture. Maybe it's not different than the control. Those are the questions that we're asking. So here we go. We're looking at organic matter. What do you see? Zero control and control, there's a slight difference. Does anybody want to guess on why there's a slight difference there? Think about it. When I came here, they were plowing in the fall and they were plowing in the spring, full conventional till. When they saw cover crops green, they're looking out there and say, I don't want to tear that up. So they stopped immediately plowing in the fall. And immediately that converts that conventional till to reduced till. Over 5 years, look at the difference from the zero control to the control. Radish, not that different from the two controls. We think that this is happening because radish has a low seeding ratio. It actually excites the microbial community. But it's not a excitement that's sustained. It's more like a jog of excitement. So think about you go to your favorite buffet. When you get there, you're excited. You just eating everything, right? And then over time, after your third trip, you're not that excited anymore. So your rate of consumption has drastically what? Decreased. That happens with radish. While it's growing, it's excited. Then it gets really excited once it winter kills, and then it warms up in the spring. And then because it's low seeding ratio, it decomposes very fast, and the residue disappears. And you know that from your experience. And then things go back to normal. Then the next radish crop, you get that excitement, termination, it gets more excited, then it goes back to normal. Now cereal rye, things hang around a little longer, higher seeding ratio. So you actually got a pretty decent increase in organic matter over 5 years. This is what you normally would see. This is what you normally would see in 10 years. And I checked these numbers. I sent samples to them, then I took them back to my lab and I did the same analysis, and we came out with similar numbers. Now I know you're looking and saying, this is 5 to 6. This ranging from what? 5.8 to maybe 7.3 or 4. What you need to know, how long has this field received manure, Russ?
50 years.
About 50 years. Before I got there. Then they stopped kind of like right when I got there. How long was this field corn on corn for silage?
Probably about the same amount of time.
50 years corn on corn for silage. This is what you get when you have that type of, and then it's a low area, so it gets wet. Okay, in wet places, decomposition is slower. So you get a buildup. Even in this rich environment, cereal rye made a dent. It made an increase, even in this rich environment. So think about, you take these same treatments, do the same study in southern Illinois, unglaciated, lighter soils, would you see some of the same trends? Possibly, okay? Active carbon, this is the carbon that's gonna be that's gonna be mineralized easily. You can see similar trend as organic matter. Okay, microbial respiration, milligrams of CO2 released per gram of soil. Look at that. These three, so we took these samples in the fall. All right, zero control, radish about the same. Cereal rye, the mixture is the highest. Can't explain the difference here, possibly that's not significantly different, but it could be that we have seen from other studies in my lab that microbially, that when you have diversity above ground, you do get more diversity below ground. So it could be that the pool is greater, so you're getting greater CO2 release because more microbes are respiring, okay? Easily mineralized proteins, okay? Milligrams per gram of soil. You can kind of see the same trend. These three are similar, and then you have cereal rye and cereal rye radish mixture kind of topping off the group, okay? So this is soil health. You know, this is soil health also, but this is really what drives everything, the microbes. And so you see an impact of cereal rye. Anything that has an inclusion rate of cereal rye is impacting the microbial respiration of easily mineralized proteins. In other words, nitrogen that's going to be released here on this slide, nitrogen that's going to be released later on in the growing season. There's more of it where you have cereal rye. Extractable phosphorus, this really shocked me. Now this field had never been, we never applied, because of the manure, we never applied phosphorus and we never applied potassium, right? When you look here, you can see they're about the same, but then here you go with the cereal rye. So the soil health test actually made this red, not because it's too low, but because it's too high, right? Because if they were on a slope, and they had a lot of water moving across, you're going to get some phosphorus releasing in erosion. But if you got a crop growing there, you're going to draw this down, and you'll be fine. You'll be right where the critical level is for this region. Potassium, you see the same thing. So I want to say to Russ, we need some potassium out in that field because we're a little low here. But potassium, you can see the difference where you have sear rot present. Okay? So those were the results of our soil health test. And again, I didn't do all of the measures. We sent them off to Cornell, and they did that assessment. And that assessment is pretty simple. It rates you from 0 to 100. The average here in this field, even with the zero control, without cover crops, was about 77, okay, compared to other soils in the region that they had tested. So the more soils that they get, the better, the more accurate that test is. But if you stay within that same test over time, you can see differences because we saw differences in treatment.